السلام عليكم أعزائي الطلبة في موضوع آخر من مواضيع أيض الشحوم، وهو أيض الشحوم المفسفرة أو الشحوم الفوسفاتية، حيث سنذكر وظائف هذا النوع من الشحوم وتصنيفها حسب الأجزاء المكونة لها، ثم نستعرض صناعة أشهر الشحوم الفوسفاتية وهو الليسيثين، ثم نتطرق إلى تحلل أو تكسير الليسيثين بتأثير إنزيمات الفوسفولايبيزز المختلفة. بعد ذلك ننتقل إلى مجموعة الشحوم المحتوية على الكحول المسمى اسفنجوسين، والتي يكون بعضا منها مفسفر مثل سفنجومايلين، والبعض الآخر منها مسكر أي يحتوي على سكر مثل السيربروسايز والجانجليوسايز، حيث سنستعرض تكون هذه الشحوم بدءا من تكون السيراميد وانتهاء بتكون السفنجوليبيد. في النهاية سنذكر الحالات المرضية المسماة سفنجوليبيدوسيس التي تحصل بسبب عدم قابلية خلايا الجسم على تكسير السفنجوليبيدز إلى مكوناتها الأصغر، فتتراكم هذه السفنجوليبيدز في الأنسجة والأعضاء ذات العلاقة. The functions of phospholipids include their essential role as constituents of cell membranes and as constituents of plasma lipoproteins, in addition to their function as lung surfactants and as emulsifying agents in the bile fluid. Some phospholipids also act as second messengers inside the cell, as well as having special functions like the role of cardiolipin in electron transport chain in the mitochondria. There are two types of phospholipids: glycerophospholipids that contain glycerol as an alcohol, and sphingolipids that contain sphingosine as an alcohol. The presence of phosphate group in phospholipids imparts amphipathic properties to them, so they are hydrophilic on one side of their structure. This is the structure of lecithin, or called phosphatidylcholine. It is a very common phospholipid, as it is present in egg yolk, milk, and for canning fatty foods. Phospholipids, such as lecithin, are a source of unsaturated fatty acids like arachidonic acid, which is released from the carbon 2 position of the phospholipid or the lecithin. So what would remain is called lysolecithin, or lecithin minus 1 fatty acid. This is the structure of a special mitochondrial phospholipid, which is cardiolipin. It is an important lipid in the mitochondria because it has a role in electron transport chain, and it is the only lipid that has antigenic properties, and this is important in some autoimmune diseases. In the synthesis of a glycerophospholipid like lecithin, the choline is first activated by ATP and then by combining cytidyl triphosphate, and then the cytidyl diphosphate choline is added to a diacylglycerol to produce lecithin. The site of synthesis of a glycerophospholipid is the smooth endoplasmic reticulum of all nucleated cells, and then the phospholipid is transported via Golgi apparatus to reach a cell membrane or secreted outside the cell by exocytosis. A glycerophospholipid can be degraded into its constituents by specific phospholipases, which are A, B, C, and phospholipase D. Phospholipase B is sometimes called phospholipase A2 and is the most important one among phospholipases, as it can release arachidonic acid from the carbon 2 position of a phospholipid. Sphingolipids contain the alcohol sphingosine instead of the alcohol glycerol. A fatty acid is attached at the amino group of this amino alcohol, while an additional compound like phosphocholine or a certain sugar is added at the primary alcohol group of the sphingosine. Look at the site of the amino group on the sphingosine and the site of the primary hydroxyl group in the sphingosine alcohol. The attachment of a fatty acid to sphingosine would form a lipid called ceramide, which can add further compounds like phosphocholine to form sphingomyelin. Addition of glucose in the form of UDP glucose would produce a cerebroside called glucosyl ceramide, while addition of galactose in the form of UDP galactose to a ceramide would produce a cerebroside called galactosyl ceramide. Addition of a branched oligosaccharide that include sialic acid or N-acetyl nuraminic acid would produce a ganglioside like GM3. This is the structure of a sialic acid or N-acetyl nuraminic acid. A sialic acid is primarily a mannosamine, a plus pyruvic acid that condense to form a 9-carbon compound, which is then N-acetylated, and it carries a negative charge. What is the importance of glycolipids? Glycolipids are seen distributed in the outer leaflet of cell membranes of various tissues, but particularly in nervous tissues. Glycolipids play a role in the intercellular interactions, and so they are important in cellular growth and development. Glycolipids serve as receptors for a variety of molecules like hormones, drugs, and even toxins of cholera and tetanus. Lastly, glycolipids are antigenic and they represent a source of blood group antigens. During degradation of sphingolipids, a defect in beta-glucosidase that removes glucose from ceramide, the disease is called Gaucher disease. In case of a defect in sphingomyelinase that removes phosphocholine from ceramide, the disease is called Niemann-Pick's disease. In case of a defect in ceramidase that removes a fatty acid from sphingosine, the disease is called Farber's disease.